So yesterday we'd gotten as far as laying out a kind of three-box model for the autonomic nervous system, and we talked a little bit about preganglionics, postganglionics, and the target. We're going to pick that idea up today, but we're going to drill down a little farther and distinguish between the parasympathetic branch and the sympathetic branch of the autonomic nervous system. Whenever you see the term parasympathetic, think rest and digest. And for our purposes today, because we're focusing on the heart, think about how we're going to decrease the heart rate. Similarly, when we think about the sympathetic branch, we think about the fight or flight response. And again, when we think about the fight or flight response in terms of the heart, we're going to increase the heart rate. So we're going to focus today on the autonomic innervation, both the sympathetic and parasympathetic branches of the heart, and how they're going to regulate the heart rate. In a little bit, probably tomorrow, we'll get into the sympathetic effects on the contractile cells. Now, one of the ideas we want to talk about here today is the anatomy and the pharmacology of the sympathetic and parasympathetic branches, and try to find those things which are in common and those things which are different between the sympathetic and parasympathetic branches. So again, we'll draw this out as a three-box model to keep it simple before we get into the nuts and bolts of the anatomy, and lay it out as a three-box model for the sympathetic and parasympathetic branches. And what we'll see is that in both the sympathetic and parasympathetic branches, the preganglionic neurons have their cell bodies in the central nervous system. And those preganglionic neurons then send processes out into the periphery. And in the peripheral nervous system, those preganglionic neurons synapse onto postganglionic neurons. True for both branches. We'll see that what's also true for both branches is that the neurotransmitter released by the preganglionic neuron is always acetylcholine. And we'll find, too, what's common for both branches is that neurotransmitter acetylcholine binds to nicotinic ionotropic channels. That is, they bind to channels that are operated by acetylcholine, and when they open, will allow sodium to flow in. So they generate excitatory postsynaptic potentials. Those excitatory postsynaptic potentials then will generate action potentials in the postganglionic neurons. And it's not really until we get to the postganglionic neurons that we begin to see pharmacologic differences between the sympathetic and parasympathetic branches. What we're going to see is that in the postganglionic sympathetic neuron, the neurotransmitter being released is going to be norepinephrine. In the postganglionic parasympathetic neuron, the neurotransmitter going to be released is going to be acetylcholine. Now, where things start to get kind of convoluted is that in the parasympathetic branch, acetylcholine is released both at the synapse between the preganglionic neuron and the postganglionic neuron, and also between the postganglionic neuron and the target. Neurotransmitter is the same. But what we see now is that it's not so much that the response is dependent upon the neurotransmitter, it's predominantly dependent upon the receptor. So where at the pre to postganglionic synapse acetylcholine is binding to nicotinic acetylcholine receptors that open channels to sodium to cause depolarization, we'll see between the postganglionic neuron and the target, acetylcholine now binds to a G-protein coupled receptor, and in a downstream effect is inhibitory. So again, it's a great example of how you could have the same neurotransmitter and have effectively opposite effects. So one of the ideas we'll have to hold on to is that when we talk about the parasympathetic branch of the autonomic nervous system, that acetylcholine is released from the postganglionic neuron, but then binds to a type of G-protein coupled receptor called a muscarinic receptor. These muscarinic receptors are simply a type of G-protein coupled receptors. Something like 4% of the genome is dedicated to making G-protein coupled receptors, and this is one subtype of those. If you take another neuropharmacology course, you'll find there's subtypes of these muscarinic receptors. We're going to talk about one type here. The muscarinic receptor binds to acetylcholine. When the muscarinic receptor, which is the G-protein coupled receptor, has bound acetylcholine, it activates the G-protein coupled receptor and now turns on a GTP binding protein. We've seen this before. Same story we saw in the endocrine system. But this particular alpha subunit is G-alpha I. You may recall that what G-alpha I does is it reduces and inhibits the activity of adenylyl cyclase. And by inhibiting the activity of adenylyl cyclase, we're now going to reduce cytoplasmic concentrations of cyclic AMP. Then that cyclic AMP, that cyclic nucleotide, is the molecule that's now going to affect the HCN channels. So if we have less cyclic nucleotide, if we have less cyclic AMP, the probability of opening those HCN channels is less. We let less sodium in. We now change the rate of rise of the ramp potential. We turn that story on its head with the sympathetic branch, where in the parasympathetic branch, the postganglionic neuron released acetylcholine. In the sympathetic branch, the postganglionic neuron releases the neurotransmitter norepinephrine. It's a catecholamine. It's a derivative of tyrosine. In the sympathetic branch, the norepinephrine binds to a particular kind of Gprotein coupled receptor called a beta-1 adrenergic receptor. The binding of the norepinephrine to this beta-1 adrenergic receptor is now going to again activate a Gprotein coupled receptor. It's going to turn on a trimeric GTP binding protein. That trimeric GTP binding protein, in this case, is going to have an alpha subunit known as alpha S, S standing for stimulatory. So in this case now, we're going to turn on adenylyl cyclase and upregulate adenylyl cyclase, thereby increasing the amount of cyclic A. So these two systems are working one against the other. We normally teach them as one being on and the other being off, the other being on and the one being on, the other being off, the other being on, and the first being off. So back and forth and back and forth. But although we teach them as being either on or off, they aren't that way. They're either a little bit on or a little bit off. There's a yin and yang, there's a balance. You can imagine this being a scale. So we're either in sympathetic drive or parasympathetic drive. When we're in parasympathetic drive, rest and digest, we're predominantly releasing acetylcholine onto the Gprotein coupled receptor and in lowering the heart rate by reducing cyclic A levels. When we're in sympathetic drive and our heart's going pitter-pat, under those circumstances, the neurotransmitter, the predominant neurotransmitter that's being released is norepinephrine that's binding to the beta-1 adrenergic receptor, upregulating the activity of adenylyl cyclase, increasing the amount of cyclic AMP, and thereby increasing heart rate. These two are constantly working one against the other. So again, an emphasis at this point. The heart has its own rate and rhythm because of those peculiar action potentials in the pacemaker cells. But because of the way the autonomic nervous system, both the sympathetic and parasympathetic branches, innervate the pacemaker cells, the autonomic nervous system now can modulate, it can regulate, it can affect the amount of cyclic nucleotide and therefore regulate the heart rate. So even though the pacemaker cells have their own intrinsic rate and rhythm, that rate and rhythm can be modulated by both the sympathetic and parasympathetic branches of the autonomic nervous system. And that's kind of the overall story we're going to try to get to today. But let's start here with our generic autonomic nervous system. And we'll do this as a kind of three-box model, again, just as we had yesterday at the close of lecture, where this is the target, this is the ganglion in the peripheral nervous system, and this is the central nervous system. This neuron has its cell body in the central nervous system and sends a process out into the periphery where it will form synapses or output regions. This neuron is known as a preganglionic. Neuron. So whether we're looking at the sympathetic or parasympathetic branches, recognize that the preganglionic neurons always have the cell bodies in the central nervous system. They send a process out into the periphery. And these preganglionic neurons synapse onto the input region of these blue neurons, whose name is the postganglionic neuron. So simply put, pre synapses onto post. Post then innervates the target. Now the target can be one of three possible structures. It could be a cardiomyocyte. The particular type of cardiomyocyte we're going to discuss today will be the pacemaker cells. We'll see later it could be smooth muscle. Could be smooth muscle of the cardiovascular system, could be smooth muscle of the gut, it could be smooth muscle, it could be smooth muscle of the gut, it could be smooth muscle of the cardiovascular system, or it could be smooth muscle of the bronchioles. It will innervate smooth muscle. And the third possible target could be a gland. And we'll discuss these a little bit later too, when we get into salivary glands and we get into sweat glands. But these are the three possible targets of the postganglionic autonomic neurons. So what else can we see that's in common? Well, the other thing that's in common here is going to be the neurotransmitter. The neurotransmitter between pre and post ganglionic neurons is always acetylcholine. So regardless of whether we're talking about the sympathetic parasympathetic branch, the preganglionics always release the neurotransmitter acetylcholine, and this neurotransmitter binds to nicotinic acetylcholine receptors on postganglionic neurons. So it's the classic arrangement. The action potentials initiated here in the central nervous system propagate along the axons, along the conductor regions, until they reach the output region where those action potentials open voltage-gated calcium channels, allow calcium to come in and trigger the release of a neurotransmitter, in this case acetylcholine. The acetylcholine binds to nicotinic acetylcholine receptors. These are ionotropic receptors, permeable to sodium ions. In other words, they're going to generate an excitatory postsynaptic potential, an EPSP. They're going to generate an EPSP in this postganglionic autonomic neuron, regardless of whether it's a sympathetic neuron or a parasympathetic neuron. That will generate action potentials now which propagate along the axon down to the target. And these postganglionic neurons synapse onto target cells. It's the same pattern. When the action potential arrives at the output region, it will open voltage-gated calcium channels. The calcium comes in, binds to synaptotagmin, goes through the snare complex, we get exocytosis, and in this case, we release neurotransmitter. What's different, however, and let's put that on the other panel, if we now draw a three-box model for the let's do the parasympathetic first. For parasympathetics, we'll now talk about this box as being the central nervous system, but in this particular case, because it's parasympathetic, it's going to be brainstem. The neuronal cell body is located in the brainstem. It sends a process to the periphery, and in the periphery, it forms a synapse in a parasympathetic ganglion. Usually, the parasympathetic ganglion for this branch of the autonomic nervous system is located in the wall of the target organ. So then this is the postganglionic neuron going out, sending a process to the target organ. Now, if you go on and take a neuroanatomy course, take some other physiology courses, in that case, what you'll emphasize with this branch of the autonomic nervous system is you'll emphasize the cranial nerves 3, 7, and 9. 3, 7, and 9. 3, 7, and 9 are going to innervate structures in the head. Some of them will go to the ciliary eye muscles, some will go to the iris, some will go to salivary glands, some will go to salivary glands, and some will go to the surface of the skin. Now, we're not going to get into that. The one we're going to talk about is the one in the brainstem located in the motor nucleus of cranial nerve 10. Cranial nerve 10 has another name. It's called the vagus. This term vagus comes out of the Latin. It means the wanderer. And it's a great name for it because these neurons are going to wander all over the thoracic and abdominal cavity and innervate organs throughout the abdominal and thoracic cavities. The one we're going to talk about here is the cardiac ganglion, of the heart. Then this neuron, which we show in blue, the postganglionic parasympathetic neuron, is now going to innervate the pacemaker cell. In the SA or AV nodes. So as above, the neurotransmitter released here is acetylcholine. And again, the receptor is the nicotinic acetylcholine receptors. If we have enough neurotransmitter bound to these NACHRs, then we generate an action potential, propagate the action potential to the output region, and at this site, we will now release the molecule acetylcholine. Again, in the parasympathetic branch, the transmitter that's released both between the preganglionic and postganglionic, as well as from the postganglionic to the target, is the neurotransmitter acetylcholine. Now, things begin to change. At this point, back here in the sympathetic ganglion, the neurotransmitter acetylcholine binds to nicotinic acetylcholine receptors, but here, acetylcholine binds to a Gprotein coupled receptor. This G-protein coupled receptor has the names called a muscarinic receptor. And again, it gets this weird name as a muscarinic receptor based on the pharmacology from way back in the early part of the 20th century. And it turns out that the mushroom from which this muscarine is taken is a mushroom called Amanita muscarina, or Amanita muscaria. And that's a mushroom that grows around here. If you go for a walk in the woods, particularly this time of year in the spring, you'll see these beautiful mushrooms. They're a red mushroom, and the cap is covered with these sort of beige-colored flecks. And you shouldn't eat them because they're poisonous. And one of the reasons they're poisonous is they contain this alkaloid, muscarine. And the muscarine actually is an alkaloid that binds to this G-protein coupled receptor. The G-protein coupled receptor here on the pacemaker cell. So this is intended to be a G-protein coupled receptor. That G-protein coupled receptor, when it binds to the acetylcholine, when it binds to the acetylcholine that has been released from this postganglionic parasympathetic neuron, it now converts the cytoplasmic structure of this G-protein coupled receptor. It changes its three-dimensional shape. When it changes its three-dimensional shape, it provides a binding site for an old friend. It provides a binding site for the alpha subunit of the GTP binding protein. We formally describe these as the trimeric or heterotrimeric GTP binding protein. You remember the story when we talked about it in the endocrine system. We said that normally when this alpha subunit and the beta subunit and the gamma subunit are all tied together, then this alpha subunit has on it a guanosine diphosphate, a GDP. But the outcome of the alpha subunit binding to the three-dimensional structure of this G-protein coupled receptor, the outcome of that is to have this GDP replaced by GTP. Let's make this a bigger cell. So this is going to be a pacemaker cell. This is a pacemaker cell innervated by a postganglionic parasympathetic neuron. So if this is our box, made a little bit bigger, and in orange we have this muscarinic, receptor, which of course is a type of G-protein coupled receptor. If it binds acetylcholine, and that now allows the alpha subunit here of the GTP binding protein to convert its GDP and exchange it for a GTP. Now, this alpha subunit with GTP bound, this is now an active alpha subunit. This alpha subunit is now going to interact with this protein. I won't draw all of them. It crosses the membrane like 24 times. This is adenylyl cyclase. Now you remember what adenylyl cyclase does. What adenylyl cyclase does is it converts adenosine triphosphate into cyclic AMP. The chemistry here is simply to take that triphosphate on ATP and cyclize it such that the phosphate is bound from the number 3 carbon to the number 5 carbon on the ribose. If you know that we make cyclic AMP, that's all we need to know. And so now that's the function of this enzyme. The function of this enzyme is to make cyclic AMP. What this alpha subunit does, because this is said to be a G alpha I, is it inhibits adenylyl cyclase. And in the process of inhibiting adenylyl cyclase, it now reduces. It reduces cytoplasmic cyclic AMP. So remember, too, that there's this other transmembrane protein in these pacemaker cells that had those 4 subunits, which we called the HCN channel. Remember that the way we drew the HCN channel was to say that it had a hole in the middle. That was the proper channel. That because it had 4 subunits, we divided it into quadrants. And we said each of these quadrants had a cyclic AMP binding site. And we said, too, that these so-called HCN channels opened when cyclic AMP is bound. But these channels are different than the other ones we talked about, because the others we talked about is whenever we bound or transfer, they open. Now, these guys aren't quite the same. They're kind of gradated over a range, so that if we bind cyclic AMP to this number 1 subunit, that increases the probability. But if we bind the second one, we increase the probability of opening even more, and by the time we bind the third, we have a pretty good probability, and by the time we get to the fourth, we're almost certainly going to have opened that channel. That's a long-winded way of saying that the probability of HCN channel opening depends on the cytoplasmic concentration of cyclic AMP. So back to this. The probability of opening these HCN channels is dependent upon the concentration of cyclic AMP in the cytoplasm. The amount of cyclic AMP we have in the cytoplasm is under the control of adenylyl cyclase. So through this pathway, through the parasympathetic branch, the release of acetylcholine from the postganglionic parasympathetic neuron binding to this Gprotein coupled receptor, we now reduce activity of adenylyl cyclase through G-alpha I. That lowers the concentration of cyclic AMP in the cytoplasm. And what's that going to do to the rate of depolarization of that membrane from these HCN channels? If you have fewer of these open, what's that going to say about the amount of sodium coming in? It's going to be less. If you have less coming in, you go up to the upper panel. Remember our graph, where this is voltage, this is time. In the pacemaker cells, we had this ramp potential. And once we reached threshold, we fired an action potential, and a ramp potential, fire an action potential, and a ramp potential. This depolarization is dependent upon sodium influx through HCN channels. So if we increase the rate of HCN channel opening, we're going to let more sodium in. That's going to make this ramp steeper. The story we just told was through the parasympathetic branch, where through the parasympathetic branch we had less cyclic nucleotide available. So that now is going to change the rate of rise such that it's shallower. Because the rate of rise of the ramp potential is shallower, it takes longer to reach threshold. So we have slowed the heart rate. So this whole process here, we're walking through a molecular explanation of why the parasympathetic branch of the autonomic nervous system slows the heart rate. And it all comes from the parasympathetic branch activating a muscarinic receptor to reduce the activity of adenylyl cyclase, to reduce the amount of cyclic A, to reduce the rate of depolarization. That's the whole story. But don't forget that this is the generic story. And this is the parasympathetic story. Now, in the backboard, let me deep six this one. And describe the sympathetic innervation of the heart. And again, we're going to do this as a three-box model, and then we can talk about the anatomy more specifically. We start out with the central nervous system. But for the sympathetic branch of the autonomic nervous system, we're now talking about the spinal cord. And we're specifically talking about the spinal cord gray matter between T1 and L3. But now we're simply replacing the box we have above, which was located in and was meant to describe the brainstem, with the spinal cord. But again, we have a preganglionic neuron. Sends a process out to the periphery. Forms a synapse in this structure here called a sympathetic ganglion. We're going to see that these sympathetic ganglia are lined up right along the spinal column. The preganglionic neuron synapses onto a postganglionic neuron, just as we saw above. And the postganglionic neuron is now destined to innervate the target, which in our case here, again, is going to be a pacemaker cell. In the SA or AB nodes. Remember that everything between pre and post is the same, regardless if we're talking about the sympathetic parasympathetic systems. And that is to say, pre to post here releases acetylcholine. The acetylcholine binds to nicotinic acetylcholine receptors. These nicotinic acetylcholine receptors are ligand-gated ionotropic channels, so again, we generate EPSPs. If we have sufficient depolarization, the membrane reaches threshold, we fire active potentials. The active potentials now propagate down to the terminal. And again, as we've seen, these active potentials will release neurotransmitter. Now, the neurotransmitter released by this post-ganglionic sympathetic neuron. This neurotransmitter is norepinephrine. Norepinephrine is sometimes known as catecholamine. It's in fact one of these molecules derived from tyrosine. Again, we have this story that the target is going to contain a G protein coupled receptor. So again, when the axon terminal, when the output region of the post-ganglionic sympathetic neuron releases neurotransmitter, releases norepinephrine, the norepinephrine is going to bind to this receptor. This receptor has a name, again. It's called a beta-1 adrenergic. You could also say norepinephrine, receptor. And now we see the same pattern reproduced all over again, where there's an alpha subunit, there's a beta subunit, and a gamma subunit. The alpha subunit has attached to it a guanosine diphosphate. When there is a three-dimensional shape resulting from the binding of the neurotransmitter to the G protein coupled receptor such that the alpha subunit can fit into that three-dimensional shape, again, same pattern, we see the GDP replaced with GTP. This GTP and the alpha subunit now turns this alpha subunit on. It activates it. This alpha subunit, in this case, is said to be G alpha S, where S is stimulatory. And specifically, it's going to be stimulatory to adenylyl cyclase. So again, we have a transmembrane protein here, and I won't put all of them in, but like 24 transmembrane regions, which is adenylyl cyclase. This alpha subunit with the GTP attached turns on this enzyme. By turning on the enzyme, we now will increase the cytoplasmic concentration of cyclic AMP. Again, the target of the cyclic AMP is the HCN channel. The cyclic AMP is going to bind to the HCN channel. If we have more cyclic AMP available, what's that going to do to the probability of opening this channel? It's going to increase the probability. If we increase the probability of, I erased it. Oh, it's buried, yeah. So now, if we increase the amount of cyclic A, what's that going to do to the rate of rise of the ramp potential? It's going to make it steeper. If it's steeper, what's that going to do to the rate at which we fire these pacemaker potentials? Make them faster. So now we can see through this autonomic innervation, the sympathetic and the parasympathetic branches, we have this ability to increase heart rate through the sympathetic system, the fight or flight system, or decrease heart rate through the parasympathetic system, the rest or digest system. And it all comes about because of the difference in the neurotransmitter released from the postganglionic sympathetic and parasympathetic neurons and the G protein coupled receptors that those neurotransmitters bind to. Yes?
If the postganglionic neuron is bound to, synapses on either the SA or the AV node, then what?
Okay, so remember the SA node and the AV node. The cells of the SA node and the AV node, those are the target cells.
Yeah, no, but like what's essentially, is that why the AV node can take over if the SA node isn't working? Like why would it synapse on both considering that the SA leads to the AV?
Okay, so this is a great question, and it speaks to an issue that's really important. Remember that these action potentials occur spontaneously. They're all dependent on the rate of rise of the ramp potential. These are intrinsic. These are the pacemaker cells that are part of the heart. They run whether or not we have any autonomic innervation. All the autonomic innervation does is modulate this. So this comes back to this idea that's sometimes hard to get your head around. That is, the heart has its own rate and rhythm. And what the nervous system does is not determine that rate of rhythm, but modulate the rate and rhythm. So what this whole story was about here with the sympathetic and parasympathetic branches was try to describe the chemical means whereby we regulate or modulate the intrinsic rate of depolarization of the pacemaker cells. Does that answer your question? Try again then.
I was just thinking, like, what's the purpose of having a synapse on the AV node if the SA node leads, like, the propagation of the action potential?
So the precise question here is why do we bother to have innervation both onto the SA node and the AV node? Well, because we want to be sure that those two the rate of rise is pretty much similar in the two. Because it's intrinsic, then if we would inhibit one more, say if we inhibited and slowed down the SA node but left the AV node intact, AV node is going to take over and we're never going to get contractions out of the SA node, or we're going to get this sort of odd arrhythmic arrangement where the atrium is contracting at the wrong time. So we need to work those two in synchrony. It's important to activate the SA node pacemaker cells in the AV node. Whether we're upregulating activity or downregulating activity, we better do it the same so that they maintain their synchronization. Okay? Yes?
In this regard, is it possible, this is just a curiosity question more in line of things, is it possible for the SA node to stop entirely?
Is it possible the SA node to stop entirely? Yes, in some cases that happens, and then they have to go in and do something, probably put a pacemaker in. The other thing that will happen, I don't get too far ahead of myself here, but the other thing that will happen sometimes is they'll in some individuals, rather than having one SA node, there'll be something known as an ectopic SA node. And there are two of them, and then they fight one another. And one of the common things that cardiovascular surgeons will do is they'll go in and ablate that second smaller node, get rid of it entirely. So you're not having this argument between these two, between the normal SA node and the ectopic SA node. And that commonly causes problems. Causes problem in people where they get tachycardia because of that. And it's an issue. And now in our modern world, you simply go in with arthroscopic surgery, find the ectopic node and freeze it usually, or ablate it one way or another. Anything else? If not, let's take the last few minutes and try to bring these boxes into some kind of anatomic reality. So we're going to take this panel and describe the anatomy of the sympathetic and the parasympathetic branches. And we're going to take the information that we have labeled on those boxes and now bring it round to a cross-sectional view and a longitudinal view of the central nervous system and the orientation and organization of the ganglia. So we can label this as the anatomic organization of the autonomic nervous system. So over here is our ice cream cone diagram of the central nervous system. This is the brainstem. There's a particular region of the brainstem known as the motor nucleus. Of cranial nerve 10, also known as the vagus. We use these terms interchangeably, whether we're talking about the motor nucleus of 10 or whether we're talking about the motor nucleus of the vagus. If we look at these, we find that it's chock full of neuronal cell bodies. It is, after all, a nucleus, and it's bilateral. And these neurons now will exit the cord, exit the brain, exit the brainstem, and go out into the vagus nerve. It's also called cranial nerve 10. Now, the ones we're going to follow are these pre-ganglionic parasympathetic for the heart. Again, this term, the vagus, indicates that this nerve is going to wander all over the place. And what we're going to do is we're going to follow this pre-ganglionic parasympathetic neuron to its output region. In the wall of the heart. It's a little shorter. In a particular region known as the cardiac ganglion. This pre-ganglionic parasympathetic neuron, as before, as we showed in the boxes, synapses onto a post-ganglionic neuron, which is now going to have a short axonal process to innervate cells in either the SA or AV node. So this is simply taking that three-box model that we've dwelt on the whole time here and bring this down to the biology of the brainstem, pre-ganglionic neuron to the cardiac ganglion to post-ganglionic neuron to target. Similarly, if we talked about so this is all, I should emphasize this is all parasympathetic. But we also said that there are a set of pre-ganglionic sympathetic neurons located in the spinal cord. And again, they're bilaterally symmetrical. And they're found between the first thoracic segment and the third lumbar segment. If we looked at this in cross-section, what we see is our classic butterfly arrangement. And there's an outpocketing here of gray matter. This is known as the intermedio lateral gray column. The intermediate lateral gray column contains all these neuronal cell bodies of the pre-ganglionic. Sympathetic neurons. if we follow these processes, they extend down into the gray matter, come out through the ventral horn, come across the ventral horn rather, come out through the ventral root, take a circuitous route, and find themselves in a structure known as the sympathetic ganglion. If we were to follow this here, we have the segmental sympathetic ganglia. And these preganglionic neurons now will form synapses in the sympathetic ganglion. There's now going to be a postganglionic sympathetic neuron, which goes from the sympathetic ganglion here all the way to the target, either in the SA or AB nodes. And now we have the sympathetic innervation of the heart. Hold on to that anatomy, and I'll show you some pretty pictures tomorrow.